Hello, good afternoon, everyone. I hope, is my audio clear?
Yes, sir.
Okay, thanks. So, I hope you all have enjoyed and had a good break. Um, let's finish off this lesson. And I think there will be 2 more classes or so through which we'll finish majority of our portion. I mean, all of it almost finished then. So we were discussing about CRISPR, uh, clustered, regularly interspersed, short palindromic repeats. In that, we need to understand several names. CRISPR is one, of course, acronym, and we also have another acronym called as Cas, that is the nuclease, that is coming from CRISPR-associated protein. There is one more acronym like PAM, that is protospacer adjacent motif. And of course, CRISPR is from here, clustered, regularly interspersed, short palindromic repeats. So there are the biology. In the last class, we focused on biology. What is their function in the bacterial genome? So it can be broken down into four steps. Their function, the function of CRISPR repeats or genes, is to provide a kind of um, immunity against bacteriophages. Right? Bacteriophages, they infect, they lyse the cell, and they go away. That means the bacteria will be killed. So their CRISPR system, they provide immunity. That means they remember which kind of phage is infecting, and then they provide specific response to that particular phage type. So it can be divided into 4 steps. That is adaptation, expression, and then interference and priming. Adaptation is the ability to pick up some DNA from the phage. Some part of the phage DNA is picked up and put it into CRISPR, the region that we have discussed previously. And the expression of CRISPR genes, the CRISPR has 2 sets. There is Cas genes and the CRISPR loci. CRISPR loci is where these protospacers are present and repeats, and Cas genes produce proteins involved in several activities. Upon transcription, it produces a pre-CRISPR RNA that is further cleaved and made into CRISPR RNAs, and the ORFs produce Cas proteins. Cas proteins are the ones that have the nuclease activity, and CRISPR RNAs are the ones that provide specificity. Specificity means where should it be cleaved. Then we have there is interference. Both these Cas proteins and these CRISPR RNAs, they form a nucleoprotein complex that is referred to as surveillance complex, which if there is any RNA or actually DNA that is complementary to the CRISPR region, then the base pair and the Cas9 protein will will cleave the DNA, thereby providing immunity or kind of protection from this particular phage. There is 4th step, priming. That is, as it is being cleaved, more and more phase sequences can be taken and put into the CRISPR region. So you might have multiple CRISPR RNAs eventually, which will be targeting the same RNA, same target sequence or the phage genome. Okay, I will quickly go through because I have discussed already. The Cas genes have 2 genes. First one is how the adaptation we have discussed. Cas1 and Cas2 are the proteins that are involved in obtaining the protospacers. Firstly, we are trying to know how are these sequences put here, the spacer here, right? To get that, the first one is called as adaptation, where Cas1 and Cas2 are involved in it. They recognize certain region called as PAM, and the adjacent sequence is picked up, processed, and then incorporated into the CRISPR array. And once upon that, there is expression and there is processing, finally generating CRISPR RNA and then CRISPR-Cas ribonucleoproteins, and then interference, as we know. So Cas1, Cas2 are not involved in interference, they are rather involved in adaptation. Adaptation is the step where the DNA from the invading phage is taken and put into CRISPR array. So there are types and other things. I'm not going to discuss that. But yeah, I think you also remember how CRISPR, the protospacer, is incorporated into the CRISPR array. I hope you remember that. It is almost same as almost similar to that of how we would have seen um, um, transposons or transposition, right? I will try to draw it like a transposition so that you probably will be able to see it that way. Let's check. Let's see. So if this is a let me draw. Sorry. So here is the assume that this is the repeat sequence somewhere around here in between. I'm not drawing it absolutely. Last time I took time to describe that. So assume that this is the middle region is the repeat. This is where the cut is made like this. And then the DNA, this target region, okay, it's going together, so it's difficult. Yeah, well, I hope you understood that there is a cut made here and that is then separated and taken into, it will become something like this then. You'll have this and a long region like this. And you'll also have, I'm now drawing these two sequences here separately. The protospacer that is brought in is now going to ligate like this here, right? The regions here, this one and this one, are actually the repeats that are indicated in here. So what happens now is that the uh, the repair mechanisms will fill up this gap, something like this. So now you previously had only one repeat, say for example, that is what is there. Now you have two repeats. Here is one and here is one. And this is the new protospacer that has been put in. Okay. Yes, that is how incorporation happens. I mean, during adaptation. So we already seen what there are two types of acquisition. Naive acquisition is the first time a piece of DNA from a phage is taken and incorporated. That is called naive acquisition. So once a naive acquisition, say one protospacer has been incorporated for a particular phage, and if that phage DNA comes again, okay, this one has come again using the CRISPR of the first acquisition, like this one. Say for example, I'm trying to draw. This DNA will be broken down, isn't it? And because it is, when it is broken down, there are more fragments formed. And if those fragments have PAM sequence nearby, those sequence can then be taken again and incorporated. That secondary acquisition of more sequences from the same phage is referred to as priming acquisition. Okay, the more number of sequences we have against one phage, the better, or the increased probability that there is more protection from that particular phage. Okay, and we also discussed about how CRISPR distinguishes self from non-self. At least, I mean, I don't know if it is completely known or not, but what you can think of is this region. So when, say, CRISPR RNA is produced, you have a tracer region, the hairpin loop of the repeat. There'll be in the target site, you will have a PAM sequence. That is fine. And then you have a target sequence. So there will be a lot of complementarity here. And the 5' side of it, if it base pairs, that means this sequence is probably, if it base pairs, then it is self. If it doesn't base pair, then it is non-self. That is one of the mechanisms, at least one of the mechanisms um, so far known, that that is how CRISPR RNA can distinguish between self and non-self. Okay. Yeah, for in today's class, we will discuss a little more about application oriented and the manipulations people have done. So it'll be a little bit biology and some of it is application. Before we go in there, I would like you to let's recollect. There is CRISPR RNA. That is the one that is RNA that is important for specificity. That is because it has homology to a target sequence. Then we have tracer RNA, which binds to the CRISPR RNA and also Cas9. Cas9 is important for the nuclease activity. It is the enzyme that cleaves the RNA or DNA. Here, mostly what we are talking about is DNA, but there are also, as I discussed earlier in one of the slides, there are sequences that are there are CRISPR-Cas systems which target RNA molecules. And then we have protospacer adjacent motif PAM sequences, which are usually present on one side of the CRISPR, the targets. This is a PAM sequence. This will also become useful or important to know because we would target. This is important because if you are trying to design or make CRISPR for a particular purpose, we should know how to choose a target and whether it is nearby a PAM or not. That's why it's important to know that as well. So we have three components that are important for the activity of CRISPR. Right, components. I'm saying PAM is part of is one of the present. It's important for activity. It is usually the part of the target here. So CRISPR RNA, tracer RNA, and Cas9. These three components together will form a ribonucleoprotein, and that is what is the actual scissors to cut the target DNA. So once people have understood the way, the biology of how it is working and so on, they have envisioned that we can do a lot of things, and that is how it has become revolutionary. So I did send a talk, or I sent a link of a talk. I hope you have seen it. Cas proteins have multiple domains. It is a huge protein, about say 1400 approximately amino acids. And there are several regions, REC1, REC2, HNH, RUVC, and PAM interacting domains. These are all the different domains, and there is a bridge helix between as well. While we are not going into all these things, it's important to know that there are two, HNH and RUVC, are important for the cleaving activity. Okay. And also PAM is directly in contact with or nearby these regions, these two domains, RUVC and HNH. Therefore, PAM interacting domain is going to influence the nuclease activity of these two domains. So if PAM interacting domain is not interacting with the PAM sequence on the target, then the nuclease activity of HNH or RUVC is not going to happen. There are more structural basis of details, but we are not going into all those things. But at least these things you should understand. The target sequence should have a PAM sequence nearby. So the CRISPR RNA binds something like this, and the Cas9 PAM interacting domain should be interacting with the PAM sequence that is present here. Okay, just briefly try to remember those things. So here is the overview we will get into. There is CRISPR locus, you have the Cas operon, you have the CRISPR array, and you have the tracer RNA. These are the three components that are required. So once they are expressed, the Cas9 and tracer, they're all involved in the, they are processed into forming, and then you will have Cas9, CRISPR RNA, and the tracer RNA, and these three will form one component. That is the surveillance complex. If the target comes, say the one that I'm showing, that is shown here, this is the ribonucleoprotein, the complex that is formed. Mature CRISPR tracer RNA, and here is the Cas9 protein. If the target is present, if the target means, target requires two things. One is there should be sufficient complementarity between the tracer RNA and the target. Second important thing is there should be a PAM sequence, which is a short GAA or something like that. It depends on the type of Cas protein. It's a very short sequence that should be interacting with the Cas protein. Okay. If you have only the complementarity is there, but no PAM, if there is no PAM, then it is not a target. A target should have sequence that is complementary and also the PAM sequence right adjacent to it. Only that is a target. Okay, keep that in mind. So because of the PAM, if the PAM is present, then the nuclease activity of RUVC and HNH is activated, and it makes a double strand breaks like this. Okay, now you have the target DNA is made into two pieces. If in the bacterial cell a phage DNA has entered and it is cut just once, even if it is cut once, it is going to be, that phage is not going to form new phage particles. Okay, so that is the danger of, that is the power of single double strand breaks. Okay, so I hope you understood what happens to the target. The target should require complementarity is one thing, and then the complementary region should be complementary to the CRISPR RNA, first point, and there should be adjacent, there should be a PAM sequence adjacent or immediately to the target sequence. Only then it is actually a target. That will activate the nuclease activities of RUVC and HNH domains of Cas9. And then the target is cleaved and the double strand break is made. And then the DNA or the phage genome is broken and it cannot replicate in the cell. That is how the cell is generally protected. So the potential is huge because the cleavage can be done. It is a double strand break, and then it is sequence specific. It's not randomly making any cuts. It is making at specific sites. It is almost similar to that of what we discussed when we started this course about restriction endonucleases. Restriction endonucleases have two things. They are nucleases and they are specific. That is about especially the restriction endonuclease of class type 2 restriction endonucleases. So there are two, if we want to commercialize and use it for our purpose, we now have to have three components. One is CRISPR, Cas9. Second is CRISPR RNA, and the third is tracer RNA. So these are the three components, right? Cas9, you have CRISPR RNA and tracer RNA. So what people have done is they, instead of making two different, they made it a single RNA, sgRNA, single guide RNA. What they basically did was, this is the CRISPR RNA, and they just made it continuous with adding a small linker loop. So it can, this is one single strand from start set here and end set here. In the previous one, if you compare, it is there are two RNA molecules. One is CRISPR RNA and the other one is tracer RNA. Now we have single guide RNA that is continuous. Only thing, it is still able to form the same structure as it was forming in the natural conditions. So that is what they have made. And now we are down from 3 components to only 2 components, right? We need Cas9 and we need a single guide RNA molecule. So it becomes much more easier or kind of simpler if you want to do it. All we need to do now is, this is the engineered one where they made it a single guide single guide RNA. You need to have an ORF for Cas9, and you need to have, sorry, you need to have a guide RNA. In the guide RNA, you will have the target sequence. Okay, you have a promoter here, and you have another promoter. And from here, you will get the Cas9 protein. It is inactive because it needs the guide RNA as well as the PAM. It needs guide RNA to start with for the activity, and specificity is given by the CRISPR. So now we have the guide RNA and CRISPR, I mean tracer and CRISPR together. So once they bind, they are active. Active in the sense for surveillance. The nuclease activity will only be activated if there is a sequence DNA with homology or the with complementarity to the targets to the CRISPR RNA here, and also there should be a PAM sequence. Okay. So that is, it is simple now. You all need to have this plasmid. Anytime you want to make a, for a specific type of target, you need to just change this one part. Then you have then you have these molecules made for the different type of target. So now we can do a lot of things, but before we go, what happens is we need to discuss a little bit of a few things. So here is a DNA molecule. Okay, assume it is a bacterial cell. It is double-stranded, and I have taken a DNA molecule. If there is a double-strand break, what will happen to it? Often, this bacterium is going to die out. Okay. Now, you also remember, I hope you also remember about what is haploid and what is diploid, right? Haploid and diploid. In haploid, you only have one set. In diploid, you have two sets. I mean, one of the reasons why there should be diploid is there is a possibility for recombinational repair, and because of which, if there is a break here, there is a way to for the second genes, the nuclear information, I mean the genetic information from the second set can be copied here. And there is always a repair mechanism also. So just try to bring back your memory about these things, and now we will discuss what can happen. So here is a DNA molecule, and if there is a break that has happened here, what are the possibilities? There are two possibilities at least, as depicted here. NHEJ, I hope you remember those. In the beginning, we have done some exercise on that. Non-homologous end joining. If you read in the repair mechanism also, you will also find that. The second one is homology driven recombination. Homologous recombination, you can put it as. Okay. In non-homologous end joining, typically what happens is there are several proteins that may process the DNA, process these ends a little more, like this and like this, and then there will be some proteins, MRX and other proteins bound here, and then they will join it together to make it a complete DNA like this. What has happened because of it? I will try to write it in terms of sequences for better understanding. Give me a minute. So I'm trying to now write a sequence: GACTAGA-T. So this is a DNA molecule. Okay. Because of CRISPR-Cas9 or any of the reasons, there is a double strand break, and the break is somewhere around here. It has broken like this. During repair, during non-homologous end joining, what happens is you may have some of the bases are usually eroded. So what happens is these things are removed like this, and they are joined. End joining happens. If it is end joining, what is the new sequence now? GAT and CTA. Okay. I think we had about 4 bases here in between, and now those 4 bases are removed. Okay. What do you think will happen because of this kind of changes? It is a mutation, and most mutations are usually non-functional, in the sense they will result in a non-functional product, or they might not have a product at all. It might be a frame shift mutation. It might be a nonsense mutation. Any kind of those mutations are likely to happen. In homologous ones, in homologous homology-driven recombination, what may happen is, assuming the same thing has happened here, okay? G, and we had some sequences here. Now it has been a little bit of processing, and there are some bases lost. But what may happen is that they will bind to, if there is homology, if they have a homology sequence in between here, and through recombination, what will happen is that it will become something like this: G and AT. And this region will get incorporated into it. Okay. So this is also a mutation, right? There was something else before, and now you have a change in nucleotide sequence, and that is a mutation. So what I'm trying to say is, through these two processes, is one way is you can delete the function of this gene that was present here, just by a single double strand break. In other case, you can do the same thing. You can create a non-functional one, or you can create what here, in this case, in non-homologous end joining, you cannot be in charge of what the sequence will be, right? You cannot decide what sequences should be there. It is a kind of random process. So we do not have that much control. But in a situation, something like based on homologous recombination, we can be in a lot of control, because the sequence given is, we gave this. If I want a particular kind of change at the end, I want a particular one type of sequence, I should have given the similar type of sequence with regions, so that they will get incorporated, and that is what it will result in. there is much more control. Okay. I hope you understood the consequences of a double strand break and how we can actually manipulate and get what we want. There are, because of these, there are many things that people have been able to do. Here, I'm going to describe a few more things. There is one called as Cas9D10A, okay, aspartic acid replaced with alanine. So they have made site-directed mutations. I hope you remember what is site-directed mutations. PCR based we have learned about how these are done, right? In that case, normal Cas9 would make it has RUVC and HNH, because of which it is able to make double strand breaks. So using site-directed mutations, they engineered a Cas9, and that is called as Cas9D10A. That means this is the original amino acid. In this position, that is 10th position, it is replaced with alanine. Aspartic acid replaced with alanine. I hope I'm right. And because of this, alanine is a non-polar amino acid, and it does. And probably this aspartic acid is important for the in the active site of RUVC. Because of replacement or substitution with alanine, the RUVC domain is no longer functional. Because of which, what is happening is it will only create one break. It can only make a single strand break, not a double strand break. Compare it with this one. It makes these double strand breaks. In Cas9D10A, it is a nickase. It can only cleave a nick, single strand break. Similarly, they also made engineered another protein called as H840A, that is histidine at 840 position, replaced or substituted with alanine. So in this case, RUVC is active, but HNH domain is not active. So it can cleave the different site on the Cas9. I mean, the target DNA, but it is also in a case, single strand break only. So there, these widen the number of possibilities that can be given. So what people have done here is something like you can also use. I'll go into the next slides. They are much more better to or easier to understand. This one is clear, right? We already discussed it. You use only one Cas9 with a CRISPR RNA, targeting only one site with one double strand break. Okay, it will either through non-homologous end joining, it will give it might mutate because of insertions and deletions at the target site, there will be a mutant formation. Or the homology directed repair, you can decide what sequence you want to replace or put into the site of the target site, and there you can allow the incorporation of the DNA that you want to. There are other ways of how you can do it. You can create, I hope you know homologous recombination. You will have to generate 3' OH by processing, and then there will be strand invasion, branch migration, and so on, and then resolution. So in that, what you do is we have made nickases, isn't it? D10A and H840A. So I hope you can see this. So here, what they do is they make one cleavage, one CRISPR is made, and here you're using one of the nickase, and you designed another CRISPR RNA to a little bit nearby, not too far away, little bit nearby, and you gave this is another nickase. So what one of the CRISPR will make is one single strand break, and another one single strand break is made like this, because of which you actually now have two DNA molecules with 3' overhangs, something like this. And they, because you're also providing a donor DNA, that is, this is going through something like HDR, homology directed recombination or repair, and the branch migration will happen, and eventually this DNA, the donor, this sequence will get incorporated into the target site. And now we have homology directed repair, and we know what sequence is incorporated in this location. Okay. Similarly, many different people have done many other things, and these are even more interesting. I think you'll see it in the next slides as well. I will repeat these multiple times, so I hope you will be able to handle with that. What if I don't want to cleave the DNA at all? Okay, the question here is, I'm not thinking about any nuclease activity. I don't want double strand break. I don't want nuclease or single strand breaks. I want to do something. I want to affect, say for example, I have an activator, sorry, a promoter. I want to repress it. How can I do it with using CRISPR-Cas? All I know is only the sequence that is present here. I know the sequence of it, and assume that the sequence also has a PAM sequence nearby. Then I can design a defective nucleus, Cas9 nucleus. When you say defective, that means it can bind to CRISPR RNA or SG, I will call it as SG RNA, single guide RNA, and it can go and bind to the target along with this, but cannot cleave the DNA. No single strands or no single strand cleavage or no double strand cleavage. So what can then it do? Now I can also add certain domains to the Cas9. I mean, this is all engineering. You make a translational fusion. I hope you understand what is translational fusion. So I take the Cas9 ORF, which is defective. So for ease of understanding, I'll put as D10A. That means aspartic acid at 10th position replaced with alanine, and histidine at 840 with alanine. So this is actually a defective one. Through site-directed mutagenesis, we can make these changes. And then we add, so I told that I want to repress here, right? My target is to repress. So I'm going to add a repressor sequence here. I know the amino acid sequence of repressor, so I'm going to fuse translational fusion. What does it mean? That means the ATG, the start codon of it starts with Cas9, and it will end after the repressor or region, reading frame, say TGA. So it will produce a protein, something like Cas9 with a repressor region here like this. And why would it come and bind here? How can I make it make the Cas9 come and bind at this particular location? That is because I know the sequence of the upstream sequence or the near the promoter region. Then I can design a CRISPR RNA or single guide RNA that has specificity or complementarity to the region with a PAM sequence that is present. I mean, the target sequence should be around a PAM sequence. And because of that, because of the RNA, CRISPR RNA or sgRNA, the Cas9, the defective Cas9 with the fused, translationally fused repressor can come here and will inhibit the transcription from this promoter. I hope you can see what all can be done. I'm just talking about repressor here. You can do the same thing as activation. You can also see, you can also visualize. That means you add a GFP protein. I want to know in a genome where exactly this particular gene, I mean, I will just give me a second. I'll make some space here. So if I have a nucleus, for example, and this is the, assume this is the nucleus, and I'm looking for, I hope you know, you might have read or not, I'm unsure, but I'll try to describe it a little bit. So the untranslated, sorry, untranscribed or transcriptionally inactive genome is usually on the periphery, and there are thought to be something like transcription hubs where there is a lot of transcription happening in one or more locations, and majority of the genes that are transcribed are usually present in that location. Okay, there is a um, nuclear architecture for in about how the genome is three-dimensionally organized within the nucleus. So if I want to know where a particular XYZ gene is, okay, if I want to know, I would have made a CRISPR RNA or sgRNA based on sequences with XYZ, and which also is adjacent to a PAM sequence. And I would have used a Cas9 defective one, okay, and I will fuse it, translationally fuse it with GFP. What would then happen if I have added this to the cell? It would go by virtue of the sgRNA, it will go and find the location where it is. And by virtue of GFP, okay, GFP will give the fluorescence. And I know that the gene that I'm looking for within the location of the gene within the nucleus is somewhere here. Okay, so it is also a good tool for visualization. I'll use more diagrams, same concept basically, and they'll probably better your understanding, so that because I'm repetitive, and also you'll see better pictures probably. Um, Here is the standard one. I'm repeating. Probably I'll stop with this one slide. Let me see if I have. Yeah, measured. All of them are the same, and I'll take last two slides. Okay. This is the standard CRISPR. Cas9, guide RNA or single guide RNA, and you have the nucleus activity HNH and RUVC. So we can also make mutants which can cleave only, which can only create nicks, and then we can make nicks in any of the places like this, cleave one on one strand, once on one strand, and on once on another strand, so that we can generate overhang. We can generate ends, DNA ends, which are much more um, ready, which can readily undergo homology directed repair. And here is one I'm trying to do: transcriptional blocking. What is transcriptional blocking? I just want the transcription to stop midway. Okay, if RNA polymerase is trying to move from here and transcribe the DNA gene, and if I synthesize if I have added a guide RNA along with a defective Cas9, it is binding here, it is obstructing, or it is kind of transcriptional interference. So RNA polymerase cannot move along because this is obstructing it. Okay. And the functional effector is the same thing as what I described in the previous slide about adding a repressor or adding an activator to stimulate or repress the transcription. So these are same things. I will probably give you the same effects how you can do it. Gene regulation, first thing, you can repress or activate by creating transcriptional fusions of these domains along with the Cas9 defective Cas9 proteins or Cas9 mutants. You can also have something like visualization, or it's called as cargo delivery. This is for visualization, right? Like GFP. If you want to locate where this particular DNA fragment or sequence is, you can use GFP translational fusions with Cas9 mutants. Here is something else, DNA demethylase. I hope you know what it is, right? DNA is methylated in the beginning somewhere. I have described about CPG islands. Those are typically present in the regulatory regions, mostly upstream of several promoters. And if the DNA is methylated, typically that region becomes silenced. So I am. I hope you remember the word epigenetics we described in some time. Change in expression patterns based on depending on any other changes other than changes in nucleotide sequence. It includes DNA methylation and histone modifications. There are other mechanisms, but at least we need to remember these two. So if I want a particular gene to be expressed and this is already methylated, how can I demethylate it? I could have just added transcriptionally fused a DNA demethylase enzyme to that of Cas9 and added a single guide RNA that is specific to region somewhere here. If it binds and Cas9 is bound, you have a demethylase which might actually remove the methylation of the DNA here, and because of which translation may happen. Transcription of this particular gene may happen. So you can influence you can make the genome do anything you want. Basically, that is how you can visualize it as. So these are important for studying gene regulation, okay, like we described. And just now I described about you want to study about epigenetic regulation, you want to control the gene regulation, you can do a lot of things using CRISPR-Cas9. What people would want to do now, however, is there is also there are also chip assay we didn't describe, or chromatin immunoprecipitation. Did we do that? Maybe not, but we will try to have a look at it in the next class. Okay. What I want you to take from this is, if we want to make a genomic change, all we need to know is the sequence. A sequence that is adjacent to with a PAM sequence. That is a target. And if I have decided that there is a sequence and PAM, and I want to know what changes exactly I want to do, I have decided everything. I want to cleave here, for example. Okay, I want to make a double strand break here. So I designed the sequence based on that. sgRNA. But sgRNA is, or the sequence of homology is between something like say 15 to 20 bases approximately. Okay. There may be somewhere else, there may be similar sequence. Now I'm not saying identical. If it is identical, it will cut here there too. There may be a similar sequence. That means there are, out of say 20 bases, here the base pairing will be 20 to 20 base pairing between the sgRNA and the target. In the similar sequences, which are referred to as off targets, that means targets that are actually not what we intend to. Out of 20, there may be somewhere around 16 or 17 homology, 17 bases matching out of 20 bases. And there is a probability that the sgRNA may bind here and Cas9 may also cut here. And this is one of the risks or limitations that we have. So it depends on how unique your target sequence is. That means there should be one and one only one sequence, and no other sequence in the genome should be very close to or similar to this to the target. If we find like that, then we have a good success rate and less of unwanted or unintended changes may happen. So there are a lot of there are multiple softwares that usually predict if you give the genome and your sequence, then the program the algorithm will look for sequence, and if that sequence is adjacent to a PAM or not. Okay, and the programs will predict. You have to give the genome if you are working with humans, mice, or whatever, or E. coli, whatever it is. You should be able to give this. You should mention it. So the program will retrieve that and will use algorithms against it to find the sequence that you are giving and see how many sequences are there nearby. And it will also predict probable off targets. So based on that, you can choose which sequence you want to go ahead with. Okay, I'll stop here, and if you have any questions, I will try to discuss. If not, I will discuss a few things again. I will review a few things again in the next class. Okay, any questions?